So previous to this mini lecture about glycolysis, make sure you've listened to and made notes on the lecture about ATP, NAD and FAD, because we're going to meet those molecules when we talk about respiration. Respiration is a complex metabolic pathway. So it's very complex. We are going to be looking at an overview of respiration. We're not going to be remembering every single stage and every single enzyme that's involved in it. What we've got on this picture here, this picture shows both aerobic and anaerobic respiration. So anaerobic respiration is the part here, anaerobic respiration, and that will be in a future mini lecture. And this is what happens when no oxygen is available. The bottom part of this diagram here, this is the aerobic respiration. So this occurs when oxygen is present, and this will release the most energy that we can produce the most ATP from to power the different reactions in our cells. And we are going to separate these stages out into 3 mini lectures. So today in this mini lecture we're going to talk about glycolysis. In the second mini lecture we'll talk about the link reaction and the Krebs cycle, and then in the third part we'll talk about the electron transport chain, which is my favorite part. One thing to note about glycolysis is glycolysis itself is anaerobic, so it is also part of the anaerobic pathway. So after glycolysis, the end product of glycolysis, pyruvate, if no oxygen is available, will enter into the anaerobic pathway. If oxygen is available, it will go into the aerobic pathway. When we're talking about respiration like this, when we talk about aerobic respiration, the overall equation we're going to be looking at is glucose, C6H12O6, plus 6 oxygen molecules will be broken down to 6 molecules of water plus 6 molecules of CO2. And in the process, we are going to release a lot of energy. And this energy will be stored in the ATP molecule that we talked about previously. So this diagram here shows the same information as the previous slide, but has the added information for location. So today we're going to talk about glycolysis, and that's going to take place in the cytosol, in the cytoplasm of the cell. We'll then have the link reaction growing into the Krebs cycle, and we'll be moving into the mitochondria. And the final stage, the electron transport chain, also takes place in the mitochondria, different location to the Krebs cycle. So it's important to look at that. If we look at the molecules that we've been talking about, so electrons are going to be carried into the electron transport chain via those coenzymes we talked about. So we'll have reduced NAD, and we'll have reduced NAD and FAD, both feeding into the electron transport chain to be oxidized. We will get ATP produced at all stages in the aerobic respiration. Glycolysis will get a little bit of ATP produced, and we call this substrate level phosphorylation. Krebs cycle is also substrate level phosphorylation. What this means is that it is ATP that is made directly by adding a phosphate to ADP. So we'll get a little bit of ATP. The most ATP is produced in the electron transport chain with the oxidation of those reduced NAD and FAD molecules, and we call this oxidative phosphorylation. We are phosphorylating or adding a phosphate group to ADP, and we're doing this powered by the oxidation of the reduced NAD and the reduced FAD. And we'll talk about that more in mini lecture 3. Aerobic respiration occurs in the cell, and glycolysis occurs in the cytoplasm. Technically, it's the cytosol. If you remember, the cytosol is the liquid part. Cytoplasm is the liquid part with the organelles in there. But this is in both animal and plant cells. So plant cells also do respiration. So not just photosynthesis. If you think about the parts of the plant, like the roots or the trunk of the tree that don't have are not green, are not going to be able to do photosynthesis. So they need to stay alive, and they can do this by respiration. So it occurs in the cytoplasm. So I'm just going to give a brief overview of glycolysis before we look at it in a bit more detail. In glycolysis, we are going to start with glucose, which is made of 6 carbons, and we were going to break that down into 2 molecules of pyruvate. And this pyruvate are each 3 carbons. So we've got C6H12O6 broken down to 2 molecules, C3H4O3. Another fact about glycolysis is that it is anaerobic, so oxygen is not needed. At the end of glycolysis, we'll have produced a net 2 molecules of ATP, and I'll tell you why I'm saying net 2 molecules towards the end of this mini lecture. Glycolysis itself is 10 different reactions, each reaction catalyzed by a different enzyme. Now, the good news is we are just looking at an overview. At this level, you do not need to remember the names, the structures of all the metabolites, those intermediate chemicals. You don't need to remember the names of all the enzymes and what they're doing. So we're very much looking at an overview and understanding what is happening. The last thing to think about in glycolysis is we said that we're going to break down glucose. The glucose needs to get into the cell. If you remember, glucose enters a cell by facilitated diffusion, so passively facilitated diffusion down its concentration gradient through the GLUT1 carrier protein, which we met in semester 1. And then once the glucose enters the cell passively through this facilitated diffusion, it will enter into glycolysis and be changed. So that means the concentration of glucose in the cell is kept very low because it is immediately changed and is no longer glucose. So we maintain the gradient, so glucose keeps entering the cell by facilitated diffusion. So you do not need to remember this picture. This is the 10 stages with the 10 different enzymes of glycolysis, and it shows you the intermediate structures and what they look like and what's occurring. So much information. But it's nice to look at some of the detail on here, even if we don't want to understand it at this level. You got the 10 enzymes. Look at the names of the enzymes. They all end in A's. So we've got hexokinase, phosphoglucoisomerase, phosphofructokinase, fructose bisphosphate aldolase, and so on. Look at this one, glyceraldehyde phosphate dehydrogenase. If you do biochemistry at university, you will be seeing these names a lot. But we've got 10 different enzymes. We just need to remember that we start with glucose, we end with pyruvate, and we need to know where we are using ATP and where ATP is being produced. I've lost where the ATP is being produced. Oh, there's the other one. I knew there was another one. And also it's good to know where we are making the reduced NAD. Okay, so they're the bits we're going to focus on. This is our simplified version of the same thing. Notice there's no chemical structures, there's not really any chemical names, there's definitely no enzyme names. This is enough. What we're going to do is I'm going to talk to you about what these little stages are doing, and we're looking at 4 main stages, but remember this is very much an overview. We start with the first stage. This is phosphorylation. So remember, phosphorylation is adding a phosphate group. And what will happen here is that a phosphate group will be added from ATP. And actually, we're going to get this happening twice. So to begin with, we're actually going to use 2 molecules of ATP. So we use 1 and we use 2. And these molecules of ATP, the energy in the bond will be used to catalyze this, and the phosphates themselves will be added to the glucose, and they will form a compound called hexobisphosphate. That's our overview name. Hexos telling us it's a 6-carbon compound. So it's got 6 carbon. The bis is for 2 phosphates. So it's going to tell us that there. And like I said, as soon as we switch glucose and stop phosphorylating it like this, it is no longer glucose. So this maintains our concentration gradient. So glucose keeps entering by facilitated diffusion through the GLUT1 carrier protein. The next stage to look at in our overview of this is stage 2. This is going to be lysis, or splitting. And what's going to happen in this stage is we are going to take our hexose bisphosphate, our 6-carbon compound with the 2 phosphate groups, and we are going to split it to 2 triose phosphates. Now remember, tri means 3. So this is showing us that we've got 1, 2, 3 carbons joined together and a phosphate group with the phosphate name. So that's what happens in the next stage. The third stage is phosphorylation again. So we're going to add another phosphate to each compound. Now this is happening twice because remember we had the 6-carbon split to 2 3-carbon compounds. So we've got 2 triose phosphates. These phosphates are not coming from ATP. Instead, they are free phosphates in the cytoplasm that are added on to the triose phosphate to form triose bisphosphate. And again, you've got that bi in there to tell you 2. The final stage in this glycolysis overview is dehydrogenation. So D means removal, so removal of hydrogen and forming of ATP. So this is what's going to occur in stage 4. So what's going to happen? We have 2 triose bisphosphates here, and each triose bisphosphate is going to be oxidized. And in the process of being oxidized, we're going to have hydrogen removed. So this is the dehydrogenation. And what's going to happen to this hydrogen is it's going to combine with NAD, which we met in our previous mini lecture, and it will form reduced NAD. And this reduced NAD will then travel off to the electron transport chain and be oxidized again and be used to produce lots and lots of ATP. In the process of oxidizing the triose bisphosphate, we are going to convert to 2 molecules of pyruvate. And if you remember, that is the end product of glycolysis. And in the process of doing this, we are going to make some ATP. And we're actually going to make 4 molecules of ATP. So you've got 1, 2, 3, and 4. So overall, we're producing 4 molecules of ATP. But if you remember, in the first stage, we actually used up 2 molecules of ATP. These 4 molecules of ATP are made directly by the phosphates of the triose phosphate being removed and combining with ATP. So we call this type of production substrate level phosphorylation. Finally, it's always really useful to keep a track of the reactants and products in these reactions. So here we've got a summary of the reactants and products of glycolysis. So we started with glucose with 6 carbons. At the end of glycolysis, we'll have 2 molecules of pyruvate. Now, these pyruvate molecules, if there's no oxygen, they'll enter into the anaerobic pathway, and we'll talk about that in a future lecture. If there is oxygen, then the pyruvate will be transported by active transport into the mitochondria, ready for the link reaction and the Krebs cycle, which we'll talk about in our next mini lecture. Remember, we're also producing 2 molecules of reduced NAD, and that again will go into the mitochondria, and that's going to be used in the electron transport chain, and that is going to produce lots and lots of ATP indirectly through a process called oxidative phosphorylation. The ATP that's been made directly in glycolysis, so we're producing 4 molecules of ATP, we call this substrate level phosphorylation. And we made 4 molecules of ATP. However, we used 2 molecules in the first stages of glycolysis. So that means the net production, 4 minus 2, is 2 molecules of ATP at the end of glycolysis. And this ATP can then be used by whatever the cell wants to use it for, to power reactions in the cell. In our next mini lecture, we are going to follow the journey of pyruvate into the mitochondria through the link reaction and Krebs cycle. Thank you.